Good morning to you guys. Good afternoon to me. This is video number 6, and our topic is photosynthesis. Photosynthesis is pretty easy if you understand cellular respiration well. So I generally teach this really hard, and then this just kind of happens. You already know that the equations of photosynthesis and cellular respiration are the reverse of each other. But it turns out the machinery or the kind of mechanics or the pathways of how it happens is also essentially the reverse. Don't forget, as far as looking at old free response, Canvas, files, AP Bio FR referenced in YouTube vids. We don't have any new ones for photosynthesis, so it's still 2019 number 3 and 2015 number 2. So there aren't any new ones. Between video 5 and video 6, it's those two. All right, so photosynthesis. Obviously this happens in plants. One common misconception is that plants do photosynthesis instead of cellular respiration. That's not true. So the idea of cellular respiration is to take food, glucose, and convert it into ATP. Plants, animals, fungus, all eukaryotes do that with mitochondria, doing glycolysis, Krebs cycle, and the electron transport. Electron transport chain, chemiosmosis is the name of the process. And so the difference is that, well, if plants do cellular respiration and we do cellular respiration, if plants do photosynthesis, what do we do instead? We eat. So plants need to find a way to make food, since they don't like gobble you up as you walk by, like you walk by a tree and or something like that. So they need to somehow get glucose. We eat glucose. They make it from the sunlight. Once we have glucose, we send it to mitochondria, cellular respiration. Once they have glucose, same thing, they send it to mitochondria, cellular respiration. So the difference is, how do you get your food? Do you eat? You're an autotroph, auto, self, troph, or troph, energy. Do you make energy yourself like a plant, or are you a heterotroph? Hetero, different. You get it from somewhere different than yourself. So I'm a heterotroph, so I eat food and then do cellular respiration. Okay, let's say that we're looking at a leaf, and so sort of like this picture right here. I look at a leaf and I look at the edge of it. I'll see tons of cells in there. Then I might zoom in on a cell, like this is a mesophyll cell. And inside of that mesophyll cell, I see all these so this is a single cell right there. I see all these green discs. Those are what kind of organelle? Yes, chloroplasts. Now I zoom in on a single chloroplast, and I see something that looks like this. I'll show you some real pics under a microscope in just a second, but essentially in the chloroplast I have these things that look like stacks of coins called thylakoids. And this is a thylakoid. So in reality, I need this whiteboard space to draw, but I'd have a thylakoid, another, but that's kind of like a coin. I'd have another, and I'd have a stack. So an individual one is a thylakoid. Knowing thylakoid is probably beneficial on the AP Bio test. You don't have to know it, but a stack of them is called grana or granum, depending on if you have a stack or stacks. Here is side by side, they show a drawing on the right, obviously, and a microscope image on the left. So I have these stacks of thylakoids under a microscope. They look like that. We're going to constantly compare mitochondria with chloroplasts. So what's the function of thylakoids looking like that? Surface area. Same thing as cristae. You might remember with cristae what I did was to increase surface area. If I had a piece of paper and I wanted to put ATP synthase and proton pumps all over it, the inner membrane, the cristae of mitochondria, and I wanted to try to fit that in my pocket, it won't fit, then my solution was to crinkle it up, and now I have increased surface area. I've gotten, an 8.5 by 6 piece of paper framed into a 4x4 pocket. Well, that's how we did that in mitochondria. We took the membrane and folded it up. Here, we take the membrane and we just make it a stack of pages like a book. Same thing, I can fit the same amount of paper in my pocket. I even gave the example of a book right here in one of the earlier videos. I've got a book. Let's say that each page is 1 square foot. It's not. This is slightly rectangular, but If that's a square foot, then that's a square foot of surface area. So it's that. And this book is 1,200 something pages long, plus about another 400 on a DVD. So this is 1,200 square feet. That's more than the square footage in this room. But I can fit it into a small space because I'm stacking it like thylakoid. So picture each one of these pages as being a thylakoid, and this is a stack, granum. Okay, so there's a thylakoid. There would be another. The next thing that we want to do is look at this and see what looks familiar. So here's my chloroplast. Here's a thylakoid. Here's a smaller drawing of my mitochondria. I know it's a little bit busy over there. What looks familiar? Well, I see ATP synthase. I already know how ATP synthase works. It's hollow, it spins. As it spins, it joins ADP and phosphate, shoves them together. And it's hollow because when protons flow through, hydrogen ions, that provides the energy to make it spin. I bet that's how this works too, and it is. Which way would protons need to flow? Would they need to flow from inside the thylakoid out into here? This area, by the way, is called the stroma. Not stoma, that's different. That's an opening so that we can bypass the cuticle of the leaf. This is stroma with an R, S-T-R-O-M-A. So this was called the matrix inside of the mitochondria. The Krebs cycle happens in the matrix. But this kind of open area where the Calvin cycle happens is called the stroma. So would the protons need to flow from the stroma into the thylakoid or from the thylakoid out into the stroma? Well, I know over here my protons needed to flow out into the matrix. They needed to go from the rectangle side to the 1, 2, 3, the 3 oval side. So same thing's going to happen here. I need a bunch of pluses inside that can flow out. And when they do, these guys will spin and make ATP. Okay, cool. Anything else look familiar? Well, these were proton pumps. That's also a purple box, so that's probably a proton pump. Hey, you know what? There's protons in there. How did they get in there? I bet the proton pump pumped them in. So, if I've got protons out here, this proton pump will push those guys inside. I guess I'll go ahead and just connect that all the way through. You'll push those guys inside. You will pump them inside. But back over here on mitochondria, how did these guys get their power to do that? Because that's active transport. When I'm eventually going from low to high, I've got a bunch of protons, not as many here. So if I'm going to go from low to high concentration, then I need energy. Where did they get their energy? Electrons. Hey, where do you think he's going to get his energy from? Electrons. These guys are new, brown with the green at the top. Those are called photosystems. Shouldn't matter in AP Bio, but technically this is called photosystem 1. They discovered it first. We also think it evolved first. And this one was discovered second, so we called it photosystem 2. This is not a pump, that's why I've got it kind of shaded. This is a docking station. I'll explain that in a second. This has the Krebs cycle, this has the Calvin cycle. Okay, so now what we want to do is compare the equations and then start getting into how this works. You already know the top one is cellular respiration. I know that because I'm breathing in oxygen, I'm breathing out CO2. If I had a glass, you might be able to see it on here. Watch this, this is just a Ziploc bag. Water vapor. Now, not a perfect example, because for all you know, that water vapor is not coming from my lungs, it just could be from like water in my mouth. But I think of it as, I breathe out CO2, and if I have like a glass, I can actually see the water vapor on it. So I also kick out water when I breathe out. I need to eat, and I need to breathe oxygen. I kick out CO2 and water, and hopefully I make a bunch of ATP. Otherwise, I'm going to die. And as we've said, I don't want to. That's a common theme in this class, not dying. Okay, the next equation is photosynthesis. We already know that they're just the equations flip-flopped, minus the ATP part, obviously. So plants, or I should say more specifically the chloroplasts in plants, because plants are they also have mitochondria up there. They take in CO2 and water, and then they convert that into glucose, which might be turned into later like fructose to make an apple. Fructose is fruit sugar, and then they kick out O2. So basically what happens is when it's winter in your area, technically like more oxygen is probably being used up, depends on the environment, but more oxygen is being used up by the environment than photosynthesis is producing. Then in the summer, O2 levels go up. You can see graphs over time. You might see these in like an environmental class as oxygen levels climb and climb and climb in the summer, then we kind of breathe it up in the winter. Climb in the summer, breathe it up in the winter. So plants, yes, they are producing O2, and yes, they are using that O2 to run their own cellular respiration, but they're producing this much and they are using that much when the sun's out. So it's almost like, yeah, I spend my own money to buy stuff for my business, but I spend $10,000 and I bring in $50,000, so I net a $40,000 profit. So as long as they are producing more O2 than they use up, that means there's O2 for you and I to breathe. Okay, so we also noticed that there's a Calvin cycle and a Krebs cycle, and basically they're going to run in the opposite way. So let's kind of look at this. Here's how I look at photosynthesis. I think that this is a very clever, simple way. I'm not saying I'm clever, but I think this is a very simple way to look at photosynthesis. If I want to build glucose, C6H12O6, then I'm going to need CHO. That's obvious. You know, I'll need 6 of those and 12 of those and 6 of those. I'll need electrons. Why do I need the electrons? What do the electrons do? Like in food, where was the energy in food contained? It was in the electrons, right? Remember how if I had like a molecule and it was held together, I had carbons, 1, 2, 3 carbons, maybe this is a pyruvate. If I could steal the electrons, so here we go, here's a pyruvate. If I could steal the electrons in the covalent bonds, the covalent bond is shared pairs of electrons. If I could steal those electrons and send them to my proton pumps, I could power those pumps and then I breathe out the CO2. Okay, well now that over here I breathed out 6 CO2s, so here's 6 CO2s floating around. What if, as a plant, I could take some of those CO2s and I could take some electrons, if I could somehow get carbons and oxygens and electrons, then I could start building glucose. I need electrons so that I can make bonds. Oops. So I can make bonds. I'm an overreactor. Quick aside. Let's say you go to Chili's. Remember back in the day when you could just go to Chili's? And for a long time they had like mugs, and I feel like they were glass. No matter what you ordered, you don't have to order beer or anything. If you just got like an ice water or a Coca-Cola, it'd be in a glass mug. So some places anyway, whether it's Chili's or not, I know they had glass mugs. Then you go to Chili's now, and it's a plastic mug and it's light. So like, you'd like lift it up, you'd kind of overreact. If I've got a drink with ice in it, and all of a sudden the ice shifts, I like way overreact. So if I think I'm gonna drop something, I overreact. It's like my body says, grab it, don't let it hit the ground or you die. And so I look like a freaking idiot. But I'm comfortable with it, so I won't edit that out. Basically, because I don't know how. Okay, so if I get all these carbons, and I can get some electrons to hold the bonds together, then eventually I can build my C6H12O6 glucose. I have 6 expo pins to represent my 6 glucoses, and then my electrons are in the bonds holding them together. Okay, so that's why I need electrons. Why do I need ATP? Because the law of entropy says if I go from disorder to order, I need energy. Or it says all things tend towards chaos, or all things tend towards disorder. They go from order to disorder, unless you input energy. So if I've got like this pile of C's and H's and O's, and then I've got electrons for bonds, but then I have to like build a glucose molecule. Remember, a glucose molecule is like a hexagon with an oxygen in the corner. If I have to organize this into the right chemical structure, that requires ATP. So if I have C's, H's, and O's, electrons to glue them all together, bonds, and then ATP so that I can organize them, I'm good to go. So in my chloroplast, the Calvin cycle is where I take all those ingredients, these ingredients, and I smash them together to make C6H12O6. The thylakoids, they call these the light-dependent reactions, and they also call the Calvin cycle over here the light-independent reactions. These depend on light. These do not. They can happen in the light, they can happen in the dark. These can happen 24 hours a day. These can only happen when there's light. So these reactions over here are generally like generating these particular ingredients, these reactants. And then the Calvin cycle puts them all together. So the analogy that I always use in class is making a pizza. Let's say that right here, this table is where I make the pizza. But, you know, the cheese is over there in the fridge, the bread is in a can on the pantry shelf, and I just got the pepperonis, so they're in like a Kroger bag still over here. So essentially there's like the step of like, I need to make the cheese, you know, I need to like purchase the cheese, purchase this, purchase this, purchase this. That's kind of like the thylakoids, the light dependent reactions. But then what I need to do is I need to ship them all to this spot. This is where I'm going to flatten out the dough and like put on the sauce and put on the cheese and put on the toppings and stuff. Pizza sounds good. So that's the Calvin cycle. This is the Calvin cycle. When I get C or H or O, I send it to Calvin. When I get electrons or ATP, I send it to Calvin, and he builds glucose, the pizza. Okay, so now let's look at this for a moment. I've got the sun, and I've got these photosystems. We call them photosystems because we run them, so to speak, or we power them with photons, light. So here comes the sun. The sunlight hits this little green guy right here. The green guys, sorry, I thought I heard something, I think it's outside. The green guys are green because those are chlorophyll molecules. Chlorophyll holds electrons in a special way such that it does this kind of complicated antenna complex and all this other stuff, but eventually the energy gets transferred to electrons, and electrons go up in energy levels. Well, you might remember when electrons go up in energy levels, they might not be stable at those energy levels. So they'll fall back down. Like we said before, if I walk outside in a dark shirt, sun hits my shirt, electrons go up in energy levels, they're not stable up there, so they fall back down and give off heat. If I'm touching my shirt when it's giving off heat, I say, that shirt is hot. Well, same thing's happening here, except when those electrons go up in energy level, absorbing energy, the chlorophyll holds them in a special way so that they don't just fall back down. So we just absorbed energy from sunlight, okay? So if I absorb energy from sunlight, then who is going to actually need it? This guy, that's a proton pump. So he could use some electrons that have been energized. So this is an electron transport chain. So now what's going to happen is the sunlight hits those chlorophyll molecules. The electrons that they're holding go up in energy levels. So here they are, they go up in energy levels. Those electrons are then transferred to this proton pump. What will that proton pump do with those electrons? They will lower them, release some of the energy from the electrons. Use that energy to pump protons into the thylakoid. What happens when I have a bunch of protons in this thylakoid and not very many out here in the stroma? They will automatically diffuse down their electrochemical gradient from high to low. When they do that, this thing will spin and we will make ATP. So if we send in ADP plus phosphate, we'll produce an ATP. So that says ADP plus phosphate. That might be kind of hard to see. So we'll produce ATP. That's good. That's one of our 3 things that we needed. I'm counting CHO as one thing. So now, where does this ATP need to go? That was one of my ingredients. Where do I send them? To the Calvin cycle. So I'll shove ATP into the Calvin cycle. The Calvin cycle is several chemical reactions, kind of like Krebs. Krebs is 9 chemical reactions. The Calvin cycle is many chemical reactions going through there, but I'm powering the Calvin cycle with ATP. I'm going against entropy with the help of ATP. Okay, so these electrons, I'll make them orange. These electrons kind of go up in energy level, whenever they get hit by the sun, they fall in energy levels as they're used on the proton pumps. How could I re-energize them? Looks like I've got another photosystem. So if the sunlight hits that photosystem, these electrons are transferred to the next one, to the next photosystem, photosystem 1, technically. They get zapped again, so they go up in energy levels again. They absorb energy again. I pass them off to this little docking station, and we're just going to leave it at docking station. It probably would be called like NADH reductase or something like that. NADP reductase, I think. And so now what's going to happen is I have to have somebody deliver those electrons to the Calvin cycle, because the Calvin cycle needs electrons. Okay, well, who were the delivery guys over here in cellular respiration? NADH, when I was full of electrons, and FADH2. Over here, almost the same thing. I have NADP plus. It'll pick up electrons, become NADPH. And it will carry those electrons. It'll be the electron carrier to send those to the Calvin cycle. I remember that because P is in photosynthesis. Over here, this doesn't have the P in it. All right, so now I've got ATP, I've delivered it to Calvin. I've got electrons, I've delivered it to Calvin. Now, where do I get my C and my O if I'm a plant? CO2, from me. So we just bring in carbon dioxide from the surrounding air. Put an arrow on it that way. Air. So from the air, we can bring in carbon dioxide. Now, you might recall that on leaves, they have a waxy layer. Wax is nonpolar. So if water is trying to evaporate from a leaf, if you figure out all the surface area of all the leaves on like an oak tree, and you figure that they're losing, when it's bright out, that those leaves would be dehydrating. So an oak tree would dehydrate really quickly. It would lose tons of water very quickly. So if I cover it with a waxy layer, water can't penetrate that layer. Right now in the winter, I know I've given this example in class, but whenever it's winter and it's dry out, water leaves my lips. My lips are not very waterproof. I have a lot of keratin in my face, right here, my cheeks, but I don't have as much keratin on my lips. That's why my lips don't look kind of, I don't know, peach or whatever color you want to call this. So anyway, when water tries to leave my lips, if I want to keep water from leaving, I just put a waxy substance on there, like chapstick or lip balm. And as long as it's nonpolar, water can't penetrate it. So water stays in my lips. Now I don't look all crusty and gross. Same thing with leaves. If I put a waxy layer on them, water can't leave. The problem is CO2 can't get in. So we have these little bitty sets of cells called guard cells, and they open up holes that will allow CO2 to move in. But whenever they open up, they're also losing water, but they're also losing oxygen, which we want to. So anyway, those holes are stoma or stomata. Oddly enough, to me, it seems like stomata should be singular and stoma is plural, but it's not. Stoma is singular, stomata is plural. The way I remember that is stomata is longer. Like stoma is a shorter word than stomata, and so stomata means many, it means more. It's plural. Take it or leave it. So that's how I get the C and the O in there. So I've got 2/3 of a check mark, like we're almost done. So I've got my ATP, thank you, ATP synthase. I've got my electrons, thank you, electron transport chain. I've got C and O, thank you, air. And now, because there's a watery environment, it turns out anytime you have H2O, I'll just go ahead and, I was going to try to do the chemical structure, I guess. I'll just write H2O. It turns out it's not always together as H2O. Water can act as a weak acid and a weak base, so it can dissociate. So some of the water, like some of the water in this glass, is OH minus ions, and some of it is H plus ions. You might see that your teachers talk about like H3O plus in a chemistry class. So H plus is available and OH minus is available. So because water is around, I have access to H pluses. So I just completed that check mark. Cool. Now when I I spin the Calvin cycle, I'll end up doing 3 CO2s. So 3 CO2s means I make a 3-carbon molecule, which is half a glucose. You likely will not have to know the name on the AP Bio test. In college you might. It's G3P, glyceraldehyde 3-phosphate. That's half a glucose. Well, what would happen if after the Calvin cycle I make half a glucose, and then I make another half glucose? What would I do with them? Shove them together to make a glucose. Okay, now let's start comparing and contrasting this. Oh, no, I forgot one thing. So what's wrong with this picture? Sunlight hits some electrons. The sunlight doesn't deliver electrons, it just delivers the energy. So just like my shirt, if my shirt is dark, the sunlight isn't delivering the dark pigments, it's just hitting the pigments in my shirt that then go up in energy levels, fall, and feel hot. So it's just sending the radiant energy. So the sun is sending radiant energy. I've got some electrons in my chlorophylls. Those electrons get excited, they absorb energy, they pass it along, here you go, the electrons are over here, the electrons end up in glucose. All right, now the sunlight hits them again, now more electrons leave and end up in glucose, now more electrons leave and end up in glucose. What's wrong with this picture? You're going to run out of electrons. Eventually all my electrons from chlorophyll are ending up in the bonds of glucose that's converted into fructose in this apple tree. So how is it that I get new electrons? From water. That's why I have to water my plants. It's not the only reason, there's also turgor pressure and all that. But I need to water my plants to give them a source of electrons. So remember how before, when back with cellular respiration, how O2 was a great electron acceptor. So at the very end of the electron transport chain, O2 would receive electrons. So it would receive a couple of electrons. So remember, oxygen just as is, it has in its inner ring, it has 2 electrons. In its second ring, it has 1, 2, 3, 4, 5, 6. So it's got a total of 8, but there's only 6, 2, 4, 6 in its valence shell. So right here, this oxygen had 8 protons because it's oxygen, and 8 electrons. So if I have 8 pluses and 8 minuses, am I neutral? Yes. But am I stable? No, oxygen needs 2 more electrons in its outer shell to become stable. So because of that, it'll form a bond and another bond with this other oxygen. Now, that functions as 2, 4, 6, 8 electrons for that oxygen. Over here, 2, 4, 6, 8 electrons for that oxygen. So now it's stable. Okay, so it needs 2 more electrons to be stable. Well, what happened was, when oxygen went in here, I have a chemical reaction, and I break these 2 bonds as I add electrons. So electrons went from here to here to here, and they ended up there, and there, there, and there. Well, now I'm oxygen. I used to have 8 protons and 8 electrons. Now I still have 8 protons because I'm oxygen, and now I have 10 electrons. So I've kind of got a 2 negative charge. This guy also has a 2 negative charge. So what was flying around in and out of these proton pumps? Protons. Now I'm back to neutral. But what did I just make? An electron and a proton. Remember, an electron and a proton make a hydrogen. 1 proton and 1 electron. That makes a hydrogen molecule. So I made a hydrogen and a hydrogen. That's water. Okay, now we're going to do the reverse over here. Over here, oxygen picked up electrons and became water. Now we're going to do the reverse. We're going to be water, drop off our electrons, and produce oxygen. So now what we're going to do is this. I'm going to do oxygen, oxygen. I'm going to use the same colors. So that's a negative, that's an electron and a proton, also known as a hydrogen. Same thing here, an electron and a proton, also known as a hydrogen. Electron and a proton, so that's a hydrogen. Electron and a proton, that's a hydrogen. So here's a oxygen and a hydrogen and a hydrogen. Remember how we were drawing our molecules on the very first video like that? That's an H, that's an H. Okay, so now we're gonna do the opposite. When this water goes up here, there's this protein. I think they still haven't isolated it. They just named it protein Z. I don't think anybody's actually found it yet. Maybe they have and I just haven't noticed on Google. But I send water up there and I've got this enzyme, we assume it's an enzyme, that's going to grab these 1, 2, 3, 4 electrons. It's going to grab these and send those along. That's where we're going to get new electrons from. So those electrons are going to leave. They're going to follow my electron transport chain and end up in the bonds of glucose. Okay, now I've got these pluses. All this happened right here. This event that I'm drawing right here is happening right here. So now these pluses, 1, 2, 3, 4, become these pluses. So they go away because they get pumped. They help me make ATP. So they're gone. Well now, this oxygen and this oxygen, they're not stable. So they need to form 2 covalent bonds. And what did they just make? O2. So then the plant will give off O2. That O2 will travel over here, pick up some more electrons, become water. That water will get sucked up by the plant, we'll steal the electrons from it, pump the protons, oxygen will be given off. This is the water cycle. This is the cycle of oxygen and water. They're just going in reverse. Now, think of it this way. The equations are in the reverse, but the mechanism is in reverse. In the Krebs cycle, aren't we ripping everything apart? In glycolysis, wasn't it like glycolysis, splitting sugar, and then I take each pyruvate and I start ripping apart electrons and everything else? What's the opposite of that? Isn't the opposite of that taking CO2s and taking electrons and shoving them together? Isn't that what the Calvin cycle is doing? It's pretty much the exact opposite of the Krebs cycle. What's the point of the Krebs cycle? Tear everything apart so that you can steal the electrons. What's the point of the Calvin cycle? Take all those parts, shove them together to make glucose. My analogy that I gave in the previous video, one of the previous 2 videos, was pretend that all I want is the glue that's holding this together. I steal the glue, the electrons, and now I just have all these pages that I don't care about, so I just get rid of them. Carbon monoxides. Well, now we're going to rebuild the book. Now we're going to take the glue and the pages and put them back together to build a book. Okay, so try this. I'm going to explain cellular respiration backwards. Meanwhile, I'm going to do my best to point over here. I want you to trace with your eyes. I want you to trace photosynthesis going forwards and see how similar it seems. All right. I start off with some oxygen I've got to do this in reverse. So water is converted into oxygen, providing electrons for some proton pumps that will pump protons, and those protons will then flow through ATP synthases to make ATP. From there, there'll be electron carriers that came from the Krebs cycle, NADH and FADH2, and they drop off their electrons. They came from the Krebs cycle. The Krebs cycle actually got 2 pyruvates. A pyruvate is basically half a glucose, and pyruvate came from glycolysis. Glycolysis began with a glucose. So they're basically reverse, right? In glycolysis, I start with glucose and I end up with 2 pyruvates, which are like half glucoses. This is not pyruvate, it's a slightly different chemical, but same idea. Here, glucose is broken in half, right? Then I send it to a cycle. Then I send it to electron transport chain. So this process is photosynthesis. If I it's not perfect, but if I do it in reverse, it's pretty much cellular respiration. Even down to the oxygen and water thing. Okay. The last thing that I wanted to look at, hang on, let me pause for 1 second. I feel like I'm missing something that I wanted to say. The last thing that I wanted to emphasize is just that, again, the Calvin cycle, I'm taking all these ingredients and shoving them together. The Krebs cycle, I'm pushing them apart. Kind of stalling. It feels like there was one other thing that I wanted to mention. I've got the water and the oxygen, got electrons. I believe that's it. One last thing. I know I always say one last thing. The thylakoid, we call these light-dependent reactions and these light-independent. That's what modern books call them. Sometimes books call these dark reactions. We shy away from that because that makes it sound like the Calvin cycle only happens in the dark. It can happen at midday in July in Texas. The Calvin cycle is just light independent. Nowhere in here do I have sunlight feeding the reaction. Over here, sunlight directly feeds these. So technically, if I could get electrons and ATP somehow, the Calvin cycle could run 24 hours a day. This part can only run when light's out. I am aware that technically, secondarily, Calvin does depend on this. So the point of the thylakoids is to produce the ingredients, the reactants for the Calvin cycle to shove everything together. I believe that's it for photosynthesis. Our next topic, we'll be looking at what the College Board releases on April the 3rd is when they're supposed to update us with stuff. So we'll be looking at what that looks like. Otherwise, our next topic is like organelles and cell communication. Thank you very much.